Hello. In this revision session, I will cover two lectures I delivered in PM2A module, and these lectures were focused on thermal analysis. So lecture number 1 was focused on thermogravimetric analysis, and lecture number 2 was about differential scanning calorimetry. So what you need to know that these two techniques are highly complementary. They measure different things, but they often could be used together to get an information about the thermal properties of different pharmaceutical materials. The first technique, thermogravimetric analysis, or TGA, it measures the weight of your sample as a function of temperature. You use a small amount of your sample, a few milligrams. You put it into the TGA instrument, into a special sample container. Then this container is sitting in an oven, and its weight is measured simultaneously by an analytical balance. And then you set up the heating regime where you heat your sample up, you increase temperature in a controlled fashion, but at a controlled speed, and then you measure how the sample weight will be changed as a function of temperature. So when your sample will start losing weight, you will see it on the TGA curve. The second technique, differential scanning calorimetry, or DSC, it works differently. It measures the difference in the amount of heat required to increase the temperature of a sample and the reference as a function of temperature. So you will have your sample always being compared with a reference, and typically the reference will be a thermally inert material. Very often it will be just empty DSC pan made of aluminium or any other thermally inert material. And again, both sample and the reference will be undergoing a controlled increase in temperature, and nothing will happen with your reference because it is thermally inert. But if there's some changes happening with your samples, then the instrument will be trying to compensate the difference between the reference and the sample, either by bringing extra heat in or taking some excessive heat out. And this compensation will be shown on the DSC curve. So thermogravimetric analysis will show the results as a weight of your sample as a function of temperature. So you could show it either as an absolute weight change, or it could show the percentage weight change. And the TGA thermograms could look different for different compounds. Some compounds will undergo single stage decomposition, and in this case they will have just a single step when the sample is losing weight in a single stage. Other compounds may be showing the multiple stages, several stages. For example, on this diagram it will be stage number 1, stage 2, and stage number 3 after the sample is almost completely lost. So it could be different scenario, different stages for different compounds. And here we have also the figure for one of the active pharmaceutical ingredients, and this active pharmaceutical ingredient was analyzed by TGA, but also, in addition to the conventional TGA representation of data using thermogravimetric analysis, it is the green curve here representing TGA. The same result could be mathematically transformed into differential form, and this differential form is called DTG, Differential Thermal Gravimetry. What DTG shows, it shows the rate of how quickly the weight of your sample is lost. So basically these two curves, they show the same thing. They show degradation, multi-stage degradation of this particular active pharmaceutical ingredient. but DTG is just mathematically transformed curve. Sometimes DTG is useful to differentiate between several thermal events that happen within a close temperature range, and sometimes it is by analyzing TGA it is difficult to differentiate several similar events, but DTG could help to do that, because DTG will, if you are losing weight, if your sample is losing weight, then you will see it better on DTG. TGA could be used for many applications, and one of the potential applications is the moisture content or moisture evaporation. So if you are analyzing your samples by TGA, then you could estimate how much moisture is present in your sample, because when water present in your sample will be evaporated, it will happen around 100 degrees. Then you will see a loss of weight for a sample. Then sample could also lose weight when it is decomposing. And at least one of the products of decomposition is volatile. Then in this case, you will be observing decomposition of your material, and sample will start losing weight. In some cases, you could also perform the analysis under so-called oxidating conditions. Typical TGA analysis is normally performed in a flow of an inert gas like nitrogen. In this case, oxidation is not supposed to happen. But then if you perform the analysis in the flow of oxygen or air, then you also could expect that oxidation may happen. And oxidation typically will, or especially oxidation in the presence of oxygen, it is when your compound will be combining with oxygen. In this case, it may gain weight. So in oxidation processes performed in air or oxygen atmosphere, you may expect that the weight of your sample increases, but this is more rare. Now let's move to differential scanning calorimetry. So this is a graph showing different events possible to observe on differential scanning calorimetry. What you need to pay attention to is this axis. Specifically, all DSC thermograms must show the direction of events. For example, in this case, you see that exothermic events are shown upwards. So all the maxima on these curves, like these two, they are exothermic, and endothermic events, they are showing downwards. So this minimum is endothermic. And typically the operator who performs DSC analysis could, before the analysis, they could set the instrument up to decide whether the exothermic events are shown upwards or downwards. So don't get confused with that. It's just a selection done by the operator of the instrument. So in this particular example, exothermic events are going upwards. Then let's go through all these thermal events we observe. In reality, you will be unlikely seeing a DSC thermogram like that for a real compound. This thermogram shows almost all possible events that you could observe, but the real thermogram will be much simpler. So what we could see on this thermogram? First, typically the differential scanning calorimetry should have a baseline, and we could see this baseline here. This will be the baseline. But also the first event, we have a baseline a bit above, and then there's a stepwise shift to the lower levels of baseline. So stepwise shifts in the baseline in DSC are typically indicating that there's a glass transition happening. So in this case, this little step is showing the glass transition, and it will correspond to the glass transition temperature. Then the next thermal event, we go in terms of the increase in temperature. The next event will be this one, this maximum, and we now know that it is exothermic event, and it could be related to crystallization, because crystallization results in release of energy, release of heat. Next event will be a minimum, which could correspond to melting, and melting is opposite to crystallization, so melting is endothermic event. It is accompanied with a consumption of energy. And then the next maximum will be the maximum corresponding to oxidation. Oxidation is the process of combining with oxidizing species like oxygen, and it always is accompanied by an exothermic event, so it is exothermic event. Again, you shouldn't expect oxidation during DSC experiment if you're running DSC in an inert atmosphere condition, in nitrogen, for example. But then if you run it in oxygen atmosphere or air atmosphere, then oxidation will be possible. Also, we have a decomposition stage here. So decomposition is the process that takes energy in, so it is endothermic, and the curve here goes down. If this experiment was continued further with further increase in temperature, what we should expect to see is that this curve eventually should go backwards and should go back to the original baseline. So what sort of information you could get from DSC, and what sort of applications are possible? First, you could use DSC for identification of different compounds, for identification of melting point, for identification of melting enthalpy. Melting enthalpy will be calculated based on the area under the melting event curve. Then you could identify glass transition temperature, and you also could identify the presence of polymorphic forms. Then you could use DSC to study recrystallization and various phase transformations. Also, you could evaluate purity. Typically, if you have a pure active pharmaceutical ingredient, that will be crystalline. In this case, we should expect that the melting peak for the pure crystalline compound will be very sharp. But then, if this melting peak will be much broader, or if it will be several melting peaks, then it may indicate that your compound is not pure. Also, DSC is often used to evaluate miscibility between different materials. Commonly, these experiments are performed for analysis of mixtures of different polymers. Amorphous polymers, they are expected to have glass transition temperatures. They will be different for each polymer, and if you start mixing two different polymers together, and if you run DSC analysis, if you see that the analysis of mixture gives you two glass transition temperatures instead of one, and these two glass transition temperatures will be roughly corresponding to the glass transition temperatures of the original materials before mixing, then this may indicate that these polymers are not mixed on a molecular level. But then if you have a single glass transition temperature from a mixture, this will indicate that the materials are miscible on a molecular level. Now let's go through several examples. So example number one. We have two diagrams. One is showing thermogravimetric analysis, and second one is DSC thermogram. So what do we see on TGA? We see that the material is thermally stable up to a certain temperature, roughly this temperature, and then it begins to lose weight, and this weight loss is almost to the very end, so material decomposes. What it indicates that this particular material will have single decomposition stage. If we look at DSC thermogram, what we need to pay attention is to direction of endothermic or exothermic events. So on this axis it does show that endothermic goes upwards. It means that exothermic will be going downwards. And what we see first is this event. And this event is a stepwise change in the baseline, which is associated with the glass transition temperature. The confirmation that it is a glass transition temperature also comes from looking at the TGA diagram, because on TGA in this temperature range we don't see any thermal events. Why is that? Because glass transition is not accompanied by a loss of sample weight. So the material just gets softer. So the first phenomenon is a glass transition temperature. Then, once the temperature increases further, we see a large endothermic event, and endothermic means the energy is consumed. And this is related to degradation. We see that to this peak on DSC, we have this massive loss of weight. This phenomena, they correspond to each other, because decomposition is the phenomenon that is accompanied, that takes energy in order to decompose, and we see this energy being taken up by a sample on DSC, and we have the confirmation from TGA that the sample decomposes. Next example. So on example number 2, what we see on TGA, we see that TGA shows us two thermal events. One happens here and another one happens there. So it's a two-stage decomposition process. Here the sample loses a bit of weight, and then at higher temperature the sample is completely burned off, so it completely disappears. And if we look at the DSC thermogram, again endothermic phenomena are shown upwards, and we also see two endothermic peaks. Each peak will correspond to decomposition of this material. Third example. In third example, we could see that the sample is thermally stable within this temperature range, and then from this temperature it starts losing weight, but the weight loss is going through several stages that are not fully clearly visible. We could have seen them a bit better if we did transform this TGA thermogram into DTG thermogram, but we could see that there's several decomposition stages up above this temperature. Now when we look at the DSC thermogram, endothermic is shown upwards, and we see this peak. This endothermic peak is not, if we compare the position of this peak, so it is observed in this temperature range, and we can't observe any weight loss in this temperature range, which indicates that this endothermic peak is not related to degradation of this compound, is not related to decomposition. What it could be related to is to melting of this compound. So we can't see melting on TGA because melting is not associated with the weight loss, but we could see melting on DSC. Then a subsequent peak is another endothermic peak here and an endothermic shoulder here. In fact, the presence of these are two different events. These indicate that these are two different events, and the TGA confirms that there's at least two different events happening. So degradation stage 1, this part, and stage 2 here, so it's not really well defined on TGA. That's why it's not like a separate peak on DSC, but it is like a shoulder of this peak. Next example. In this example, what we could see that the TGA shows us a bit of stability initially, but then the mass of sample goes upwards, which means that the weight is not lost in this case, but it is gained. And the gain in weight in TGA could only be associated with oxidation. And oxidation is typically exothermic event. And if we look at DSC, endotherms they are showing upwards, exotherms showing downwards. And we clearly could see that this peak is exothermic, very broad exothermic peak associated with oxidation process. So once the oxidation is completed, the sample is stable in terms of its weight within this temperature range, and then it suddenly starts to lose weight, so it degrades. And that's what we see. We see that the subsequent DSC diagram shows us the endothermic series of events that are of endothermic nature. Example number 5, well, it does show that nothing is happened both in TGA and DSC. It means that this particular sample, when it was analyzed, it doesn't show any thermal events in a given temperature range. Sample is completely stable and it doesn't show any melting or any other events. I hope you find this revision session useful. Again, if you have any questions, then feel free to drop me an email. I will try to answer all your questions. But also, what you could do, you could practice in interpretation of different types of analysis, either TGA or DSC, by simply trying to Google DSC of any drug you could come up with. For example, if you will type in Google DSC of ibuprofen, and then you will go through Google Images, then you will see lots of thermograms for different materials. Maybe not all of them will represent ibuprofen or any other material, but you could go through different examples, and you may find some papers where they provide some interpretation of different types of analysis. And the same could be done with TGA. In fact, in many papers you will see that researchers have analyzed one compound or mixtures of compounds by several different techniques, and that will give you some more examples of interpretation, and that will solidify your knowledge of the thermal analysis methods. Good luck with your exams.